Welcome back for another deep dive. This time, we're going to zoom in, way in,
Mm.
to the world of nerves.
Microscopic level.
Yeah, those tiny electrical wires that basically make our bodies, well, work. Imagine, you accidentally touch a hot stove,
Ow!
right? You pull your hand away, like instantly, without even thinking. We're going to break down what's happening there, the amazing chain reaction in your nerves that makes that possible, and we'll be referencing a fascinating physiology handout by Dr. Rasha Eldeeb. It really gets into the structure and function of nerves.
It's amazing how these tiny structures control everything we do. Feeling a breeze on your skin,
Yeah.
or coordinating complex movements, playing the piano, hitting a tennis ball.
Right. So much complexity packed in there. Let's start with the basics. What's a fundamental unit of this system? The nervous system.
The neuron,
Yeah.
the building block. Think of them as specialized messengers. They transmit information throughout the body in the form of electrical signals
Right. And chemical signals.
and chemical signals.
Okay. So neurons are like the communication network of our bodies.
It's exactly.
Relaying information from our brains to our muscles, senses to our brains.
Yeah, everywhere. And these messages travel along nerve fibers. Dr. Eldeeb mentions two main types, myelinated and unmyelinated.
Myelinated and unmyelinated. So is that what determines the speed of the signal?
You got it. Think of myelin like insulation, like the plastic coating around a wire.
Oh, okay.
It helps speed up signal transmission.
So myelinated nerves are like the express lanes.
Yeah.
Speeding
Yeah.
things up.
Yeah.
And unmyelinated.
They still get the job done, just at a slower pace, like taking the scenic route. You'll get there. But it takes a bit longer. In the central nervous system, fun fact, the myelin sheath is formed by these special cells, oligodendrocytes.
Oligodendrocytes, that's a mouthful.
Yeah,
But I guess even the nervous system needs its support staff.
absolutely. It's a team effort.
Mhm.
Now, to really understand how nerves work, we need to look at their special properties. One of the big ones is excitability,
Okay.
their ability to respond to different stimuli,
Stimuli. Like what? Give me an example.
like heat, touch, chemicals. Remember that hot stove?
Yeah.
The heat, that's the stimulus that triggers your nerves.
Right. So it's like our nerves are these sensors picking up information and reacting to the world around us.
Precisely. And once a nerve is stimulated, it has to transmit that signal. That's conductivity. Nerves can transmit signals along their length, like
Right.
a chain reaction, carrying information from one part of the body to another.
Okay, so excitability is like the on switch.
Mm.
And conductivity is how the message actually gets delivered.
Right.
And there's also something about this all or nothing thing with nerves. What is that?
The all or none law, it's pretty straightforward. Nerves either fire a full-strength signal or nothing at all, no in-between. Another interesting thing, they don't tire out easily. This is called infatigability, meaning they can keep firing signals without getting exhausted.
Wow. So it's like they have unlimited energy. Imagine if your nerves needed a coffee break. Right.
We'd be in trouble.
We would be. Now, the effectiveness of a stimulus, meaning how well it triggers a nerve.
Depends on two things, the strength of the stimulus and how long it lasts. The duration. Dr. Eldeeb mentions the strength duration curve.
Oh, the strength duration curve. That sounds complex. What is that exactly?
It shows the relationship between these two, the strength and the duration.
So a strong stimulus doesn't need to last as long to trigger a nerve, right?
Right. And a weaker stimulus needs a longer duration to do the same. Like think about pushing a heavy door.
Okay, yeah.
You can either push really hard for a short time or push with less force but for longer. Either way, the door opens.
Makes sense. Now, I see some terms here in the handout. Rheobase, utilization time, and chronaxy. These all sound very technical.
Yeah, they're related to the strength duration curve.
Okay, so can you break those down for me?
Sure. Rheobase is the minimum strength of a stimulus needed to excite a nerve at all. Utilization time is how long a rheobase stimulus needs to last to be effective. And chronaxy, that's the duration needed for a stimulus twice the strength of rheobase to trigger a nerve.
Okay, it's a lot to keep track of. But essentially, these measurements help us understand how excitable a nerve is.
Exactly. And this has real-world applications. Like in physical therapy, you can use that knowledge, understanding chronaxy, to figure out the best duration for electrical stimulation to activate specific nerves.
That's pretty amazing. So by studying these properties, we can actually learn to better target and stimulate nerves for therapeutic purposes.
Exactly. And all this talk about stimuli and nerve responses, it brings us to the heart of nerve function, the action potential.
Okay, the action potential.
Yes.
This is where it gets really interesting. So the action potential is that electrical signal that travels along the nerve fiber, right?
You got it. But before we get into that, We need to understand something called resting membrane potential. It's the electrical charge difference between the inside and outside of a nerve cell at rest. Think of it like a battery, storing potential energy.
Oh, okay. So the inside of a nerve cell has a negative charge compared to the outside, like the two poles of a battery.
Exactly. And when a stimulus comes along, that's when depolarization happens. The stimulus causes the nerve cell membrane to become less negative, and eventually it flips, becomes positive. It's like a wave of positive charge sweeping along the nerve.
So it's like the battery discharges its energy.
Yeah. And after the signal is sent, the nerve cell needs to reset. This is repolarization. The nerve cell membrane goes back to its negative resting state, ready for the next stimulus.
So it's like recharging the battery.
Exactly. And all of this is powered by the movement of ions, sodium and potassium ions. They're the key players in this electrical dance.
Okay. I'm starting to see the bigger picture. We've got the neuron, the nerve fiber, the stimuli, and the action potential, that electrical signal that makes it all happen.
Right.
But I imagine a nerve can't just keep firing and firing. It needs a break.
You're right. After an action potential, there's this period called the absolute refractory period, where the nerve can't fire another signal no matter what.
So like a mandatory timeout.
Exactly. Like when you take a picture with a flash, you have to wait for it to recharge.
Right.
Then there's the relative refractory period, where the nerve can fire again. But it needs a stronger stimulus than usual.
So there's a kind of cool down period.
Exactly. And remember, all sorts of things can affect nerve excitability. Temperature, pressure, blood supply, oxygen, even chemicals.
Like our nerves are super sensitive to their environment.
Absolutely. Now that we know how individual nerve cells work, let's talk about how those impulses travel along the fiber. Think of it as the nerve superhighway.
Okay. That sounds interesting. And I bet it's different for myelinated and unmyelinated nerves.
You bet it is. In unmyelinated fibers, the signal moves like a wave, a continuous wave.
So smooth and steady.
Yeah, but in myelinated fibers, something amazing happens. The signal jumps between the gaps in the myelin sheath. This is called saltatory conduction.
So it's like taking the express train and skipping all the local stops.
Exactly. So much faster.
Wow. The myelin really does make a huge difference.
It's the difference between sending a message by carrier pigeon or email.
Okay, before we move on to what happens once these signals reach their destination, there was one more thing in the handout.
Yes.
Electrotonic potentials.
Right.
What are those exactly?
They're localized changes in the nerve electrical potential, but they happen in response to stimuli that aren't strong enough to cause a full action potential.
So like a little nudge, but not enough to make it fire.
Exactly. And they play a role in fine-tuning nerve activity. It's like they're tipping the scales, making it more or less likely that the neuron will actually fire a full action potential.
So they're like little whispers of activity that can either amplify or dampen the signal.
Exactly. And there are two main types, anelectrotonic and catalectrotonic potentials.
Okay, those names sound a little intimidating.
Yeah.
What's the difference between them?
Okay, so imagine you're trying to push a heavy object. An anelectrotonic potential, it's like someone's adding a bit more weight, making it harder to get it moving. It actually decreases nerve excitability. But a catalectrotonic potential, that's like someone giving you a little push, making it easier to get the object going, increases excitability.
Oh, okay. I get it now. So it's like fine-tuning the system, making sure those nerves are ready to respond in different situations.
Exactly. Now, shifting gears a bit, let's talk about action potentials. You might be surprised to learn they're not all the same.
Wait, really? I thought an action potential was just an electrical signal. How can they be different?
Well, it depends on how we're actually recording and measuring that electrical activity. Dr. Eldeeb's handout describes a few different types. Let's start with biphasic action potentials. These are what we see when we've got two electrodes placed on the outside of a nerve fiber.
So we're listening in on the electrical conversation happening inside the nerve, but from the outside.
Exactly. As the action potential travels along the nerve, it first reaches one electrode. So you see an upward deflection on the recording. Then, as it passes between the two electrodes, the potential difference becomes zero, so there's a downward deflection. And finally, when the signal hits the second electrode, another upward deflection.
So it creates this up-down-up pattern as the signal moves along. I'm picturing it like a boat going by.
Yeah.
As the front passes you, it creates a wave going up. Then as it moves past, the water level goes back down. And as the back passes, another wave going up.
Perfect analogy. Now, there's also something called a monophasic action potential. This one is recorded when an electrode is inside the nerve fiber and the other is outside. or sometimes when an electrode is placed over a damaged part of the nerve.
So with a monophasic recording, we'd only see a single upward deflection as the signal passes.
Exactly. And then there are compound action potentials. This is what we see when we record from a mixed nerve, a nerve that has different types of fibers with different sizes.
So instead of a single clean peak, we might see a more complex waveform with multiple peaks, kind of like listening to a choir instead of a soloist. Each fiber is contributing to the overall sound.
I like that. And speaking of different types of fibers, Dr. Eldeeb's handout also mentions a way to classify mammalian nerve fibers based on their diameter and how fast they conduct signals.
Okay, tell me more.
There are three main groups, A, B, and C. Group A fibers, these are the big ones, largest diameters, fastest conduction speeds. Think of them like the high-speed internet cables of the nervous system, handling things like motor control, sensory perception, and reflexes.
So these are the ones that let us react quickly to the world around us, like dodging that tennis ball or pulling our hand away from a hot stove.
We got it. Then we have Group B fibers, a bit smaller, a bit slower than Group A, but still important. They transmit signals in the autonomic nervous system. That controls things like your heart rate, digestion, breathing.
Okay, so they're more like the steady, reliable phone lines, keeping those essential functions running smoothly.
Exactly. And finally, we have the Group C fibers, the smallest and the slowest. They're involved in transmitting pain and temperature sensations.
Interesting. So even though they're slow, they're carrying some really important information, like alerting us to danger.
Exactly. It's fascinating, right? Our body prioritizes different types of information based on how quickly it needs to get there.
That's amazing. So it's like our bodies have a built-in system for managing information flow, prioritizing the urgent stuff.
Precisely. And here's another key takeaway. Each fiber type is good at its specific job, but that specialization also makes them vulnerable to different things. Like Group A fibers are sensitive to pressure, while Group B are vulnerable to lack of oxygen.
So different conditions can affect different parts of the nervous system in very specific ways. It's like this complex web of pathways, each with its own strengths and weaknesses.
Exactly. It really shows the incredible complexity of our neural circuitry and how adaptable it is.
This has been so insightful. I'm learning so much about how nerves work, from the basic building blocks to the action potentials. But before we wrap up, one question popped into my head. We've talked about how nerves respond to stimuli. What happens next? How does the brain actually make sense of all these signals? How does it decide what to do?
That's the million-dollar question. That takes us beyond individual neurons and into the brain itself. It's like we've been looking at the hardware, and now we need to understand the software, the algorithms and processes that make sense of all this electrical activity.
So we've learned the alphabet, but now we need to learn how to put the letters together to form words and sentences.
Exactly. That's where it gets really interesting, because we're talking about perception, cognition, consciousness.
Wow, that's a whole other deep dive. But before we get too carried away with the brain, anything else we need to cover about how nerves work?
Actually, yes. There's one more thing I wanted to mention. It has to do with these different types of nerve fibers and their roles in transmitting information.
Okay, I'm all ears. So we've talked about these different types of action potentials and nerve fibers, but you said there's more to it when it comes to how they actually transmit information.
Yeah, Dr. Eldeeb's handout highlights this idea that our nerves are organized in a way that prioritizes certain types of information, almost like a highway system, with different lanes for different speeds.
Oh, interesting. So some signals get the express route, others are taking the scenic route.
Exactly. Think about it. You touch a hot stove. That pain signal needs to get to your brain, like, instantly.
Right, so you can pull away.
Right. But information about, like, subtle pressure changes when you're holding a pen, that doesn't need to travel quite as fast.
Makes sense.
Mm.
But how does our body actually decide which signals get priority?
Well, those different fiber types we talked about, A, B, and C, remember, they play a big role. Those large myelinated A fibers, they're like the express lanes, carrying signals for things like motor control, touch.
Proprioception.
Proprioception, that's a big word.
It's your sense of body position,
Yeah.
where your body is in space. Oh, okay. So those are the signals that need to be super fast, so we can react quickly and move smoothly.
Exactly. Then you have the smaller, slower B fibers, important for the autonomic nervous system, controlling things like heart rate, digestion, breathing.
Okay, so those are vital functions, but they don't need to be lightning fast.
Right. More like a reliable but leisurely schedule.
Gotcha.
And then finally, the C fibers, those unmyelinated fibers carrying pain, temperature, and itch signals.
So even though they're the slowest, they're still carrying super important information, like warning signals.
Exactly. It's like an alarm system. Might be a bit slower, but still vital for keeping you safe. This whole organization. It makes sure our nervous system can handle a ton of information efficiently and prioritize the critical stuff for immediate action.
That's incredible. Our bodies have this whole intricate system for managing information.
It really is. And honestly, we've only scratched the surface of this whole field. But hopefully, this deep dive has given you a new appreciation for just how complex your nervous system is.
It definitely has. I never realized how much is going on behind the scenes, even for simple things. Neurons firing, different signals racing around. It's mind-blowing.
It is. And before we go, I wanted to leave you with one last thought. We talked about how nerves transmit signals, the brain interprets them. But what about glial cells?
Glial cells, yeah, I've heard of them. But I don't really know much about them. Aren't they just kind of like support cells for the neurons?
For a long time, scientists thought that too, that they were just passive. But now we're learning they play a much bigger role.
Really? Like what?
Well, they're involved in regulating synaptic transmission, how signals are passed between neurons, they control blood flow in the brain, they even influence the formation of new connections between neurons.
Wow. So they're not just bystanders after all.
Not at all. They're like essential partners to the neurons, helping to orchestrate all that brain activity.
That's fascinating. I'm definitely going to have to read more about glial cells. They sound like the unsung heroes of the nervous system.
Definitely worth looking into. It's a hot area of research. Tons of exciting discoveries happening all the time.
Well, I'll add that to my list of deep dive topics. Thanks for taking us on this journey into the world of nerves.
My pleasure. Keep those synapses firing and never stop exploring the wonders of your own brain.
And to all our listeners, thanks for joining us for this electrifying deep dive. We hope you learned something new and exciting about the amazing world of nerves. Until next time, keep diving deep.